Welcome everyone. In this chapter, we are going to study nucleic acid. Nucleic acids are very important molecules because they store and transfer genetic information. The two main nucleic acids are DNA and RNA. Today we will understand what they are made of, how they are formed, and what their function are. Before we start, let us see what we will cover in this chapter. First, we will learn the definition of nucleic acid. Then we will study the nitrogen pages called purine and pyrimidine. After that, we will learn the component of nucleoside and nucleotide which is assembled. Then we will study the structure of DNA using the Watson-Crick double helix model. Finally, we will learn about RNA and the function of different type of RNA. Nucleic acid, which are DNA and RNA, are formed by the joining many small units called nucleotide. So we can say nucleic acid are polymers and nucleotide are their building blocks. Each nucleotide has three parts: a sugar, a nitrogen base, and a phosphate group. DNA and RNA are the main molecules that carry genetic information in living cells. They play a very important role in controlling life processing, especially protein synthesis. Because proteins are produced based on genetic information. Historically, nucleic acids were discovered in 1869 by a Swiss scientist named Friedrich Meyer, who isolated a new substance from the nucleus of white blood cells. The discovery was the beginning of nucleic acid research. Now let us understand the structure of a nucleotide chain. When a DNA or RNA is completely hydrolyzed, it gives three compounds: first a bundle sugar, second phosphoric acid, and third nitrogen containing heterocyclic compound and called nitrogen base. In the actual DNA or RNA chain, nucleotide join together and after another to form a long chain. The sugar and phosphate from the backbone of the chain, and the nitrogen pages are attached to the sugar and project outward. Now we study the first component, which the pentose sugar. Pentose sugar means a sugar with 5 carbon atoms. In DNA molecule, the sugar is beta-D2 deoxyribose. In RNA molecule, the sugar is beta-D-ribose. The main difference is simple. Ribose has an OH group at the 2 carbon, but deoxyribose does not have that occasion. Because of this difference, DNA is called deoxyribose nucleic acid and RNA is called ribonucleic acid. Now we study the second compound, which is phosphoric acid, written as H3PO4. The phosphate group is very important because it links nucleotides together. When one nucleotide connected to another nucleotide, the connection happens through the phosphate group. Phosphate group also give nucleic acid their acidic nature and give negative charge to DNA and RNA. That is why nucleic acid behave as acidic molecule. The third compound, which is the nitrogen pages. Nitrogen pages are two main type of purine and primetin. Purine are double ring structure and primetin are single ring structure. The purine are adenine and guanine. The primetin are cytosine, thymine and uracil. Cytosine is common in both DNA and RNA. Thymine is present mainly in DNA, while uracil is present mainly in RNA. This point is very important for exams. This slide supports the idea of purine and primetin. The key point to understand is that these nitrogen pages are like letters of the genetic code. The order of bases in DNA decide the genetic information. Later, RNA reads this information and help in producing protein. So the nitrogen bases are essential for storing, drink and transferring information in body. Now we look again at the nitrogen bases. Purine and adenine and guanine. Primetin are cytosine, thymine and uracil. A simple way to remember is DNA has ACCD and RNA has ACCU. So thymine is replaced by uracil in RNA. This difference helps us identify DNA and RNA exams. Now we learn formation of nucleoside and nucleotide. First, let us understand nucleoside. When a bento sugar combines with a nitrogen base, it forms a nucleoside. This happens when the nitrogen base is attached to the one carbon of the sugar. Next, when a phosphate group is added to the nucleoside, it becomes a nucleoside. So in one simple line, sugar plus base is equal to nucleoside and nucleoside plus phosphate is equal to nucleotide. This is a very common exam question, so please remember to clearly. Now we see example is DNA. In DNA, sugar is deoxyribose. So nucleosides are called deoxynucleosides. For example, deoxyadenosine is formed by adenine plus deoxyribose. Deoxyguaninine is formed by guanine plus deoxyribose. Deoxycytidine is formed by cytosine plus deoxyribose. Deoxythymidine is formed by thymine plus deoxyribose. These are the basic nucleosides found in DNA. Now we study the formation and the structure of nucleotide. A nucleotide has three compounds: nitrogen base, pentose sugar, and a phosphate group. In RNA, the sugar is ribose. In DNA, the sugar is deoxyribose. The phosphate group attached to the sugar and the nitrogen base also attached to the sugar. So the nucleotide structure of differ slightly between DNA and RNA mainly because of the sugar and one base difference, thymine versus uracil. Now we understand nucleotide formation more clearly. When a phosphate group linked to the 5-OH, a nucleoside through a phosphoester linkage, a nucleotide is formation. RNA nucleotide include adenyl acid, guanylyl acid, cytosolic acid, and uracilic acid. DNA nucleotide include deoxyadenylate acid, deoxyribonucleic acid, deoxyribonucleic acid, and the deoxythioaminonucleic acid. These nucleotides join together and form long chain of DNA and RNA. Now we start the most important part, the structure of DNA. The Watson-Crick model is also called as the double helix model. DNA was first identified as acidic substance present in the nucleus by Friedrich Messier in 1869. She named its nuclein later. Watson and Crick proposed the correct double helix structure. This structure explained how DNA can store genetic information and how it can replicate during cell division. Now we learn the main advantage of the double helix structure. DNA is made of two polynucleotide chains. These two chains run in opposite directions, which is called antiparallel arrangement. The pages present in the two strands pair with each other by hydrogen bonding. The page pairing is specific. Adenine pairs with diamine and guanine pairs with cytosine. Because of page pairing, DNA remains stable and also can copy accurately during their replication. This slide shows DNA. DNA is genetic material in most organisms. DNA is present mainly in nucleus in eukaryotic cell. DNA carries genes and genes decide characteristics and control cell activity. DNA itself does not directly make protein, but it provides instruction through RNA. Now we study function of DNA. The first function is genetic information transfer. DNA carries genetic information from parent to children. The second function is replication. DNA makes a step copy of itself during cell division so that new cells get same genetic information. The third function is transcription. When RNA is formed from DNA, itself called transcription. This RNA then helps in protein synthesis. So DNA is the master control molecule for retrieval and protein production. Now we move to RNA, which is ribonucleic acid. RNA is present in all living cells and has similarity to DNA, but it is most often single-stranded. RNA contains ribose sugar and bases A, C, C, and U. RNA is mainly involved in protein synthesis. In simple words, DNA stores the message and the RNA helps to use the message to make protein. Now we see the bases in RNA. Attached to the sugar is one of four bases: adenine, uracil, cytosine, or guanine. Different type of RNA exist in cell. The main types are mRNA, dRNA, and rRNA. These three work together during protein synthesis. So RNA is not only one type. It has different forms with different roles. Now we study the function of different RNA types. mRNA is messenger RNA. It carries a genetic message from DNA to ribosome and helps in protein synthesis. dRNA is transfer RNA. It brings amino acid to the ribosome according to the code written in mRNA. RNA in ribosomal RNA. RNA form a major part of ribosome and it supports the assembly for amino acid into the protein. So overall RNA control and support protein synthesis by carrying the message, bringing even more amino acid and forming the ribosome structure.